Hello, my name is Delaney Rickles, and I'm a high school senior from Marietta, Georgia. Today, I'm going to be giving you a crash course on fluorescent microscopy, which is, I think, one of the most fascinating things in science. All of the scientific materials you see used in this video were filmed in a biomedical engineering lab where I currently intern. Fluorescent microscopy is a way of looking at microscopic particles by looking at the light that they emit, rather than through the traditional way of shining light onto them. Samples are stained with different types of fluorophores, which are fluorescent compounds. The fluorophore used depends on what the scientist is examining. To understand how fluorophores emit light in a fluorescent microscope, we must go back to some basic chemistry principles. Electrons are located on different shells of atoms. When light is shined on fluorophore electrons, they become excited and they naturally want to jump to electron shells closer to the nucleus of the atom. As the electrons jump up shell levels, they grow more and more excited until they release the extra energy they are holding and fall back to a lower shell. The energy released reaches us in the form of light. You may remember that light is not just the white-yellow light bulb that we see. It's an entire spectrum of wavelengths, and certain wavelengths excite fluorophore electrons more so than do others. So how does a fluorescent microscope excite fluorophore electrons, and what's the purpose of doing so? As I mentioned before, each type of fluorophore is excited most by a specific wavelength of light. By staining samples with fluorophores designed to detect certain aspects of a cell, we can identify whether those targeted areas are truly present. For example, if I stain a sample with DAPI fluorophores, I can tell if nuclei are present because their image will appear via the DAPI stain. Fluorescent microscopes have an amazing way of executing this complex process. First, light enters and is filtered to the optimum wavelength for the fluorophore stain being used. This occurs through an excitation filter. This light excites the fluorophores and they release light in return. The light emitted by the fluorophore electrons has a different wavelength than the light that excites them. To ensure that we only see the light emitted by the sample, another filter is put in place beneath the microscope's eyepiece. Here we can see a turntable which changes the excitation filter on a real fluorescent microscope. Although the filter is contained inside the microscope, we can easily observe the change by observing the filters act on samples in the dark. In this video, I am changing the excitation filter from a wavelength which best activates DAPI stain to a wavelength that best activates FITSI stain. All of this work culminates in beautiful pictures of cells, tissues, and other samples that scientists use fluorescent microscopy to observe. Thank you so much for watching my video. If you're a beginner, I hope you learned a little something about fluorescent microscopy, and if you're a Science Ambassador Scholarship Judge, I thank you for your consideration and thank you for the opportunity.